Environmental mutagens include constituents of smoke, paints, petrochemicals, pesticides, dyes, foodstuffs, drugs. The principal means of exposure are inhalation, skin absorption, and ingestion. Mutagens such as nitrates and nitrates are converted into mutagens by body chemistry. So, today we are going to discuss mutagenesis and DNA repair. Let's discuss different types of mutagens, which are base analogues, chemical modifiers, and intercalating agents. Let's discuss base analogues. Well, base analogues cause transitional mutation. Transitional mutation causes substitution of one purine with the other purine, like adenine to guanine, or guanine to adenine, or substitution of one pyrimidine with another pyrimidine, such as thymine to cytosine, or cytosine to thymine. Let's take few examples to understand the mechanism of base analogues and alternations caused after its incorporation. Well, 5-bromo-uracil is a common example of base analogue, which shows analogy with nitrogenous bases like thymine and cytosine. 5-bromo-uracil exists in two states. One is normal keto form, which is thymine, and it can be converted to the other. Rare enol form, which is cytosine, through tautomeric shift. Thus, the base analogues show tautomerism. Tautomerism means they exist in two interconvertible forms. Moving forward, we know that adenine forms bond with thymine, but this thymine, through substitution, converts to 5-bromo-uracil. It is normal keto form. Then, first round of replication occurs. With this adenine, thymine bonds, and to this keto form of 5-bromo-uracil, adenine bonds. Then, second round of replication occurs. With this adenine, thymine bonds, but keto form of 5-bromo-uracil, through tautomeric shift, converts to enol form of 5-bromo-uracil, which is cytosine. Then it forms bond with guanine. Then third round of replication occurs, and with this guanine, cytosine bonds. Enol form of 5-bromo-uracil converts to keto form of 5-bromo-uracil, which is thymine, and forms bond with adenine. That's why base analogues cause transitional mutation, because parent strand had adenine and thymine, but in daughter, transition of adenine to guanine and thymine to cytosine has occurred. Moving to the next example of base analogue. Similarly, 2-aminopurine is another example of base analogue, which show analogy with nitrogenous bases like adenine and guanine. It exists in two states. One is a normal amino form, which is adenine, and thus the base analogues show tautomerism. It means they exist in two interconvertible forms. Normal amino form, which is adenine, converts to other, which is a rare iminoform, that is guanine, thymine, bonds with adenine, but this adenine, through substitution, converts to 2-aminopurine. It is normal aminoform. Then first round of replication occurs. To this thymine, adenine bonds, and to this aminoform of 2-aminopurine, which acts as adenine, bonds to thymine. Then second round of replication occurs. To this thymine, adenine bonds, but aminoform of 2-aminopurine through apotymeric shift converts to iminoform of 2-aminopurine, which acts as guanine and bonds with cytosine. Then third round of replication occurs, and to this cytosine, guanine bonds, iminoform of 2-aminopurine converts to aminoform of 2-aminopurine, which acts as adenine and forms bond with thymine. That's why base analogues cause transitional mutation, because parent strand had thymine and adenine, but in daughter, transition of thymine to cytosine and adenine to guanine has occurred. Next mutagens are the chemical modifiers. Example of chemical modifiers is nitrous acid. Nitrous acid converts cytosine to uracil and adenine to hypoxanthine. Hypoxanthine is a precursor of guanine. Alkylating agents modify bases by donating alkyl groups. Let's understand it. Thymine bonds with adenine. Nitrous acid deaminates adenine into hypoxanthine. Thymine forms bond with adenine. Hypoxanthine forms complementary pairing with cytosine in place of thymine. Hypoxanthine is a precursor of guanine and forms bond with cytosine. So, in parent stand, there was thymine and adenine, but in daughter, transition of thymine to cytosine and adenine to guanine occurs. Moving to next mutagen, and that is intercalating agents. These include acridine dyes, proflavin, and ethidium bromide. Let's understand that. How they act as a mutagens. This is DNA sequence. Acridine orange acts on it. Acridine orange possess air, hydrophobic heterocyclic ring. It binds with the DNA major and minor grooves. Intercalating agents also mimic the nucleobases and intercalate between the DNA nucleotide bases. It results in atypical spacing between the nucleotides and distorts the DNA helix. The unusual spacing in the DNA helix causes the insertion or deletion of bases in a growing DNA chain. As a result, the normal reading frame of the bases changes, thereby causing frameshift mutation and ultimately leading to aberrant DNA replication and transcription. Now, let's discuss safety measures when using X-rays. Patients' previous X-ray exposure should be reviewed before requesting further X-rays. Next is the 28-day rule. A woman of childbearing age should be X-rayed only if she has had a period within the last 28 days. Next is the 10-day rule. For high radiation dose procedures, such as abdominal or pelvic computed tomography and barium enema, the examination should be postponed to the first 10 days of the menstrual cycle. Other are electromagnetic radiation. Particulate discharge from radioactive decay includes alpha particles, called as helium nuclei, beta particles, called as electrons, and gamma rays. The mutagenicity of subatomic particles depends on their speed, mass, and electric charge. The energy and mutagenicity of electromagnetic radiation increases with decreasing wavelength. All radiation beyond ultraviolet causes ionization by knocking electrons out of their orbits. Other are ultraviolet light. Ultraviolet light is the non-visible, short wavelength fraction of sunlight responsible for tanning. It exerts a mutagenic effect by causing dimerization of adjacent pyrimidine residues, mainly thymine, but also thymic cytosine and cytosine. It does not cause germline mutations, but is a major cause of skin cancer. Other are atomic radiation, includes natural background radiation, cosmic rays, man-made radiation, and X-rays. Now let's discuss mechanisms for DNA repair. These are direct repair, Base-excision repair, Nucleotide Excision Repair, Mismatch Repair, and Post-Replication Repair. Well, DNA repair, apart from direct repair, all the DNA repair systems in human cells require exonucleases, endonucleases, polymerases, and ligases. Let's discuss direct repair. Well, a specific enzyme reverses the damage. For example, by dealkylation of alkylated guanine. Let's understand it. DNA sequence with alkylated guanine. Now, specific dealkylating enzyme acts, and as a result of which guanine is restored. Next is the base-excision repair. The damaged base, plus a few others on either side, are removed by cutting the sugar phosphate backbone, and the gap filled by resynthesis directed by the intact strand. Now let's understand it. The DNA sequence has uracil, created by deamination of cytosine, causes mismatch with guanine in next step. It is cut by uracil DNA glycosylase, then endonuclease in next step. It is replaced by DNA polymerase, and finally sealed by ligase. Next is the Nucleotide Excision Repair. This removes thymine dimers and chemically modified bases over a longer stretch of DNA. Defects cause xeroderma pigmentosum. Let's understand it. This is a DNA sequence. Thymine dimer are produced due to UV exposure. Then, in next step, removal by endonuclease. In next step, replaced by DNA polymerase. And finally, sealed by ligase. Next is the Mismatch repair. This corrects base mismatches due to errors at DNA replication and involves nicking the faulty strand at the nearest guanine adenine cytosine and thymine base sequence. Defects occur in hereditary non-polyposis colon cancer. The genes involved are sometimes called mutator genes. Let's understand it. This is DNA sequence. Guanine adenine thymine and cytosine endonuclease. Cuts here. There is base mismatch, which are cytosine and adenine. In next step, exonuclease removes bases through error. In next step, replacement by polymerase. Correct base inserted. And finally, sealed by ligase. Last is the post-replication repair. This corrects double-strand breaks either unguided or by using the homologous chromosome as a template. Defects include Bloom syndrome, which is a chromosome breakage syndrome. Other are breast cancer and possibly prostate cancer. Let's understand it. In first step, double strand break. In second step, there is creation of single strand tails. In third step, there is invasion of homologous chromosome by single strand tail. Fourth step is repair synthesis. And finally, repair completed. So, it was all. Well, hope you have a better understanding of mutagenesis and DNA repair. Thank you for learning. Join us again.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.